Now we are going to look at animation of gel electrophoresis. So let's look at that animation. Now we are going to talk about the gel electrophoresis procedure. It is used to separate DNA fragments of different sizes. As I've mentioned, electrophoresis uses electric current to separate different size molecules in a porous sponge-like matrix. Just the same principles apply to molecules going through narrow spaces as they apply to people or vehicles trying to make their way through a traffic jam. The particle, smaller the entity, the quicker it will move. Just like in a traffic jam, a person on foot can move faster than the one on the bike, which can move faster than the one on the motorcycle, or which can move faster than the person traveling in a car, so on and so forth. The smaller the size of DNA, the quicker it will move through the gel. The gel is basically a matrix through which these DNA molecules have to move. It is placing obstacles in the path of DNA, so it is restricting the movement of DNA. The smaller the DNA, the quicker it can move. I would like to mention that one of the people who invented this procedure, Dr. Bill Sugden, he was also my professor while I was studying at University of Wisconsin, Madison. He's one of the people who invented this procedure. As I've mentioned, it is made from, agarose gel is made from purified seaweed, and we can pour it like a gel we use at home and make it any shape we want. And in this case, we are making it a rectangular shape, and we have placed a comb at a specific location. One end of the gel had a comb, and when we remove the comb, the place where the teeth of the comb were, they basically form little wells in which we can add our DNA sample. Then we take this gel and place it in a container and fill it with a buffer that can conduct electricity. So after we have done that, the samples will be loaded in the gel. So here we are going to load the samples. Here's a pipette which is loading different samples. Remember, one of these samples will also be the known size DNA fragments. These known size DNA fragments will be used to compare the size of the DNA fragment that we have produced with PCR. Here we are pointing out that DNA is negatively charged molecule, and it will move towards the positive electrode once the electric current is applied. And the region between the well and the tip of the other tip of the gel is basically the lane. So this is basically the lane in which the DNA molecules will move. Since the shape of the well is rectangular, squished rectangle, that would basically also be the shape of the band. So now let's move forward. We are going to apply the electric current. The DNA is going to move. Remember, the gel is sort of like a lump of sort of accumulation of mesh of hair, and the smaller the DNA, the quicker it will move towards the positive electrode. So here, when we turn on the current, the DNA in the well starts moving towards the positive electrode. So after we have run our gel, we will stain it with the special dye. This dye binds to DNA. Generally, people have been using ethidium bromide. It's a flat molecule that can insert itself inside the space between the spaces, or interspaces between the bases, and when the stained gel is exposed to ultraviolet light, the DNA bands start to glow. Here, marker is basically, or the DNA ladder it is also called, because it forms like the steps of ladder. These DNA fragments of different size. Say for example, this may be 500 base pairs, this may be 800, this may be 900, 1000, 2000, 2500, 3000, 3500, so on and so forth. So when we run the gel, we place our PCR product and run it, we can compare, we can estimate the size of our PCR product. Here in this gel, we are not actually loading the PCR products, it's a different product, but the principle still applies. We can look at the size of the DNA fragments that are present in the gel. This gel can also be photographed, and the photograph can be stored or filed in for future references. So here we have a procedure which allows us to detect the product of PCR reaction, and also very importantly, we can measure, we can have some rough idea about the size of our PCR product. The procedure we looked at is the gel electrophoresis.